there's been an explosion of interesting research over the last few years. I guess I would have to start with the development of the mRNA vaccines and the technology for the rapid development of vaccines that we've benefited from as a society in the fight against the COVID pandemic. Also, it's been really amazing to see the growth in cell-based therapies, and gene-based therapies are not far behind. A very exciting promise for the future. There's been many advances in biomedical engineering as well that are really exciting in many different areas. And we've also seen, in a very gratifying way, this expansion of interest in resolution biology and the science underlying that and potential for therapeutics in this scientific space, especially in the fight against more chronic inflammation. Hypothetically, the answer would be yes, it's an area of great interest scientifically. We don't have clear answers yet. We know that long COVID is associated with increased severity of acute COVID. And what has been learned is that lipid mediators like cytokines during acute severe COVID are dysregulated. It's been described as a cytokine storm and a lipid mediator storm. And so in that context, we've seen dysregulation of resolution mediators and mechanisms. And so it is quite possible that people that are suffering from long COVID, in part, may have resolution deficits. But that's an area of active research for now. Cytokines are generally released from cells in response to a challenge, often an inflammatory or infectious challenge, but it could be tissue injury as well. And those cytokines are released from those resident cells of the injured or inflamed tissue to excite the local cells, but also to recruit and activate cells that are part of the acute inflammatory response. In disease or disordered settings, their production becomes out of step with an organized, orchestrated inflammatory response that's self-limited. And so in COVID, it was such an alarm signal, and what happened is that the cells, principally in the airway and in the lung, started releasing cytokines in very high levels and in a way that created almost excessive, you might call it excessive inflammation that had a long tail to it. So the cytokine storm was really the production of an array of these inflammatory mediators, these signals that are being released. And it's almost a call for help. And the troops came roaring in to the point where the inflammatory response actually contributed to the severity of the initial COVID infection. So that's how I would describe a cytokine storm. In many experimental systems, in animal systems, in translational human systems, experimental systems, what we've seen is potent bioactivity for external SPMs with minimal toxicity. And again, this has now been demonstrated in early human translational research studies as well. So there's quite exciting and significant potential for external SPMs to be used in a therapeutic manner. So how best to think about that? There are a couple different ways I think about this. One is we all have a bit of a set point, if you will, a homeostatic set point for inflammation, and that can be influenced by internal things like genetics, other diseases, but it can also be influenced by external things like your diet, like the climate, like pollution exposures, etc., other environmental exposures. And so external SPMs have the scientific potential to be able to reset that inflammatory threshold, if you will, and help in a protective manner from inflammatory challenges or infections. But also, we know that resolution biology seems most active countering inflammatory challenge. And so in a post-acute sense, after somebody develops an infection, an injury, or an inflammatory challenge, in that post-acute phase, accelerating a more timely resolution of that inflammatory challenge. So this is very relevant in the setting of exacerbations of underlying disease. Asthma is one of the most common ones, for example. But also in diseases of chronic inflammation, where you're post-acute and you're in now the more subacute or even chronic phase, is an area where there's exciting potential for external SPMs. Related to the mechanism of action, it's very cell type dependent and context dependent. We know that the external SPMs have the potential to influence cell responses from tissue resident cells, airway lining cells, fibroblasts, other macrophages, but also infiltrating leukocytes, so circulating cells as well. So that answer, I think, is more complex. And so the mechanism of action typically is receptor mediated in the context I'm thinking about, but it's also cell type and context dependent. So there are many examples where the mechanism has been elucidated, but in humans in vivo, I think it's too complex to be able to predict a singular mechanism of action. So asthma is the most common chronic condition in humans in the airway that's inflammatory. Maybe 1 in 10 to 12 individuals in the United States are affected with asthma at some point in their life. So very common, and it's a chronic inflammatory disease of the airway where we don't currently have any disease-modifying therapies to actually cure asthma. But there are other chronic inflammatory diseases that are common. One might be psoriasis on the skin. There are types of inflammatory arthritis that we can see in the musculoskeletal systems, like rheumatoid arthritis, for example. There are many other examples as well. Inflammatory bowel disease, yet another. So here at the 17th annual Bioactive Lipid Mediators meeting, we're presenting work on specific cystinyl SPMs, so SPM conjugates, and how they're influencing lung inflammation, lung infection from important respiratory infections. And also we're trying to understand, and so we're presenting data on how these SPMs, these external SPMs, signal to regulate cell function. And so we've identified an intracellular signaling mechanism for the SPMs and how they can regulate cell function. So we're presenting data on all of those things. More generally, we're very interested in roles for SPMs, internal or external, in regulating lung inflammation and resilience in response to challenge. And we've had a significant focus recently on acute models of inflammation, and we're broadening that out to more chronic models now. And then we're also looking in a translational mechanism at long COVID as a potential for a deficit in resolution, but also asthma as a potential model system for disease-modifying therapy. And we're participating in large national NIH-funded efforts along those lines to translate the work from the basic science and model systems in animals to now human health and disease.